Hello, my name is Joshua Mugabe. I welcome you to Coordination and Control, questions 36 to 40. Question 36. State the functions of the major plant hormones and their site of production. B. Describe the mechanism by which auxins bring about cell elongation in plants. Part C. Some birds remain dormant throughout the dry season. Why is this so? State the functions of the major plant hormones, their site of production. We have auxin, iodoacetic acid. These are produced at the apical meristem, other immature parts of the plants. They promote stem elongation and growth, formation of adventitious roots, inhibition of leaf abscission, promotion of cell division, that is with cytokinins, inducements of ethylene production, and promotion of latelobud dormancy. We have hormone cytokinins. This is produced from the root apical meristem and immature fruits. It stimulates cell division, but only in the presence of auxin. Promotion of chloroplast development, delay of leaf aging, and promotion of bud formation. Another hormone is gibberellins. This is produced from the root and the shoot tips, young leaves, and seeds. It promotes stem elongation of young. It stimulates enzyme production, that is in leaves and in germinating seeds. We have ethylene. This is produced from the roots, shoot, apical meristems, leaf nodes, edging, flowerings, and ripening fruits. They participate in the control of leaf, fruit, and flower abscission, as well as promoting ripening of the fruits. We have abscisic acid. This is produced from the leaves, fruits, root caps, and seeds. ABA, it participates in inhibition of bud growth, control of stomata closure, some control seed dormancy, and inhibition of the effects of other hormones. Brasinosteroids. These are produced from pollen, immature seeds, shoots, and leaves. Have overlapping functions with auxins and gibberellins. Oligosucklins. They are produced from cell walls. They participate in pathogen defense and reproductive development. Part B. Describe the mechanism by which auxins bring about cell elongation in plants. We have our structure here trying to show us how the auxins bring about cell elongation. This is the cell. We shall cut a smaller piece, and that smaller piece will be enlarged to give us this structure here. So we have here the cytoplasm of the cytoplasm, and we have the cell membrane here, and then lastly outside we have the cell wall. And in the cytoplasm we have the auxins, and then we have the hydrogen ions. Then we have in the cell wall, we have the enzymes which are inactive, and then we also have the cross bridges. Those are the cross bridges in the fibers. The auxin stimulate the release of hydrogen ions from the target cells, which alters the pH of the plant cell wall. This optimizes the activity of the enzymes which break the bonds in the cell wall, allowing them to expand. So if we look roughly here, we have in our cytoplasm. In the cytoplasm, that's where we have the auxins, and these ones are going to stimulate the hydrogen ions in this target cell, and these ones of course will enter into the cell wall. When they enter in the cell wall, they are going to alter the pH of the cell wall. When they alter the pH of the cell wall, they are going to activate the enzymes which were inactive, and these enzymes are going to break the cross wall, the cross bridges. And when the cross bridges are broken by the enzymes, they are going now to cause the expansion of the cell wall. The cell wall is going to expand, and by expanding as they allow in more water to enter. Part C. Some birds remain dormant throughout the dry season. Why is this so? The lateral bud inhibition or apical dominance, which is likely controlled by auxins, The gradient of the auxins and gibberellins from the apical buds attract food and cytokinins. The food substances, for example sucrose, from the leaf is translocated past the dormant lateral buds, which have high concentration of the growth inhibitor like abscisic acid. The lateral buds are inhibited and they become dormant. (37) Each of the following events requires the recognition of a molecule. Explain how each of the following recognition process occurs. Part A. Target cells recognizing the hormones. Part B. B cells recognizing the antigens. Part C. Postsynaptic membranes recognizing the neurotransmitters. Let's look at the target cells recognizing the hormones. There are two ways in which the target cell recognizes hormones. In cells that recognize steroid hormones, the hormone will diffuse through the plasma membrane and nuclear envelope and binds with the special receptor proteins in the nucleus. These receptors then activate transcription of specific genes. If the hormone is a peptide, the hormone binds to a special receptor protein on the plasma membrane surface. These receptors in turn, they trigger the conversion of ATP to AMP, which in turn activates an enzyme that produces a particular cellular function. B cells recognize antigens. The B cells have special antigen receptors on their plasma membrane. In the production of B cells, millions of possible genetic combinations produce B cells with different kinds of receptors that will bind to different antigens. When an antigen enters the body, it will bind only to the B cell with the specific antigen receptor. When this occurs, the B cell proliferates, producing plasma cells that have exactly the same antigen receptors. The antigen receptors are then released into the body fluids as antibodies, which bind and inactivate the antigens which stimulated their production. Part C. Postsynaptic membrane recognize neurotransmitter. When a neurotransmitter, such as acetylcholine, is secreted by the presynaptic membrane, diffuses across the synaptic cleft to the postsynaptic membrane. The neurotransmitter binds to one of the many specific receptor proteins in the postsynaptic membrane. Depending on the neurotransmitter, the type of postsynaptic membrane, the binding of the neurotransmitter on the receptor will cause the influx of sodium ions or an efflux of potassium ions, resulting in an excitatory postsynaptic potential or an inhibitory postsynaptic potential. Briefly explain the effects of hypo and hyperthyroidism in humans. Part B. How are the secretions of the thyroid gland controlled? Briefly explain the effect of hypo and hyperthyroidism in humans. We shall start with the hypothyroidism. Primarily hypothyroidism. This is characterized by cold intolerance, mental retardation, obesity, low heart rate, somnolence, low basal metabolic rate, and many other. Cretinism in infancy, characterized by sluggishness, physical and mental retardation, dystrophy of bones, soft parts, and lowered basal metabolic rate. Myxedema, especially in adults, it is characterized by the generalized body swelling, mental or physical sluggishness, reduced metabolic rates, reduced heartbeat rates, lowered body temperatures, and obesity. Hypothyroidic goiter. This is characterized by thyroid glands swelling due to compensatory rise in thyroid activity in response to hypothyroidic state. Let us now look at hyperthyroidism. Primarily hyperthyroidism. It leads to increased basometabolic rate, increased heartbeat rate and ventilation rate, raised body temperature, heat intolerance, restlessness, muscle wasting, easy fatigability, and other features of heart failure. Thyrotoxicosis, thyroid storm. This is a fatal condition of hyperthyroidism due to excessive secretion of thyroid hormone, characterized by sudden onset of palpitations, confusion, heart failure, raised blood pressure. Exophthalmic goiter, goiter accompanied by protrusion of the eyeballs. Part B. How are the secretions of the thyroid glands controlled? Thyroxine level are controlled by negative feedback mechanism. Low basometabolic rate, meaning low thyroxine level in the bloodstream. This triggers the release of thyroid releasing hormone, TRH, from the hypothalamus, a hormone that is sensitive to the thyrotropes of the anterior pituitary gland to release thyrotropin, thyroid stimulating hormone, TSR. The thyroid stimulating releasing hormone then stimulates the follicular cells of the thyroid gland to release thyroxine, tetraiodothyroxine, a hormone that raises the basometabolic rate. Very high basometabolic rate, that is high thyroxine level, switches off the thyroid releasing hormone secretion, and the thyroid stimulating hormone also ceases. Thyroid gland thus stops further secretion of thyroxine till normal is re-established. So when you look here in our graph, you can see that, for example, when the environmental temperatures are varying, the hypothalamus is going to detect that. And when it detects that, it is going to release a releasing factor, which will go to the pituitary gland, and then the pituitary gland will be caused to secrete the thyroid stimulating hormone, thyrotropin, which will move to the thyroid gland and causes the thyroid gland to secrete the thyroxine. Because when the thyroxine is produced, there is increased in basometabolic rate, and there will be now a negative feedback inhibition going to the pituitary gland, preventing it to release the thyroid stimulating hormone, and then also going to the hypothalamus, preventing it from releasing the thyroid releasing hormone. And then the thyroid gland will stop secreting thyroxine. Thyroxine also directly inhibits further release of thyroid releasing hormone and consequently thyroid stimulating hormone. (39) With appropriate examples, explain the concept of correlation in plant growth and state how it has been utilized in horticulture. Part B. How does the morphology of an etiolated plant suit it for growing through soil? Part C. How are the various tropic responses important to the plant? Part A. With appropriate examples, explain the concept of correlation in plant growth and state how it has been utilized in horticulture. Correlation is a process by which one part of the plant controls another via the influence of the growth substances. Classical example is the apical dominancy, which is a phenomenon whereby the presence of a growing apical bud inhibits the growth of the lateral buds. Apical dominancy is under the influence of auxins. It also includes suppression of later root growth by the growth of the main root. The removal of the shoot results in the lateral bud growth, branching. This has been applied in pruning when bushy rather than tall plants are required, for example, in tea farms. Part B. How does the morphology of an etiolated plant suit it for growing through the soil? The hooked plumule of the dicots protects the delicate apical meristem in form of soil particle. The elongated internodes ensure maximum chance of reaching light. The small leaves offer less resistance to the passage through the soil. Leaves the grass remain inside the coleoptile. Part C. How are the various tropic responses important to the plant? Positive phototropic and negative geotropic responses of the shoot and coleoptile allow exposure of the leaves to sunlight for effective photosynthesis. Root negative phototropic and positive geotropic allow the penetration of soil. Rhizomes and runners are geotropic. Helps the plant colonize near areas of the soil. Hyphae are positively chemotrophic, allow growth towards food. Tendrils are positive haptotropic, essential for support. Sundew tentacles are positive haptotropic. They enable to capture insect prey. Lateral roots are plagiogeotropic, allow exploitation of large volumes of soil as well as providing support. Branches are plagiotropic, maximize volume of space for effective capture of light. Pollen tube are positive chemotropic for growth towards the ovule, allowing successful fertilization. Pollen tubes are negatively aerotropic, allows growth of the pollen tube towards the style away from air. Dicot leaves are diagiotropic, maximize exposure of the leaves to maximize capture of light at a right angle of incidence radiation. Roots and pollen tubes are positive hydrotropic, allow access to water, vital for various living processes. Number 40, outline the physiological and biochemical effects of glucocorticoid like cortisol. Part B, how are the secretions of glucocorticoids like cortisol controlled? Part A outlined the physiological and biochemical effects of glucocorticoid like cortisol. Carbohydrate metabolism. This promotes gluconeogenesis. It promotes liver glycogen formation, raises blood glucose level. Protein metabolism. It promotes the breakdown of plasma proteins to yield energy, increases the availability of amino acids for enzyme synthesis in the liver. Other roles. Prevents inflammatory and allergic reactions. Decreases antibody production. Part B. How are the secretions of glucocorticoid like cortisol controlled? Cortisol is controlled by negative feedback. Low levels of glucose in the bloodstream trigger the release of corticotropin releasing hormone from the hypothalamus, which also stimulates the corticotroph cells of the anterior pituitary gland to release adrenocorticotrophic hormones, which causes the adrenal gland to release cortisone from the adrenal cortex, which accelerates gluconeogenesis and glycogenolysis. Excess glucose in the blood stream inhibits further release of corticotrophic releasing hormone from the hypothalamus, which also inhibits release of adrenocorticotrophic hormone, suppressing further release of cortisol from the adrenal cortex. Thank you very much for participating in coordination and control. Questions 36 to 40. Please do like, share, and subscribe.